Teeth. Teeth are amazing. They help you chew, have a great smile, keep your jaw in place, and even retain the bone in your jaw. But from the millions and millions of cells in your body, how is one cell supposed to differentiate into a tooth cell? Here's our friend, Jerry. His incisor grows throughout his life, and this is done with the help of epithelial cells that create ameloblasts. Ameloblasts help create enamel, which is the outer layer of the tooth. Enamel isn't grown evenly on the tooth, but instead prefers to grow on the labial surface, meaning towards the lip. The epithelial stem cells that help control the growth of Jerry's incisors are found in niches called the labial and lingual cervical loops, or CL. This is seen near the root of the tooth. But between the two, the labial CL seems to make the ameloblast. There are different types of BMPs, or bone morphogenic proteins, that are expressed in different stages of the developing tooth. When noggin overexpression disrupts BMP activity, molar development and late-stage incisor epithelial cell differentiation are blocked. But if there is a lack of noggin, then the upper incisors can fuse. Jerry wouldn't have a very pretty smile if this happened. Also, if the specific BMP receptor, BMPRLA, is inactivated, tooth development is halted. Before we continue, you are probably wondering what noggin is. Well, it is a BMP antagonist and is also a direct target of microRNA 200C. So microRNA 200C and BMP signaling work in a positive feedback loop. Also, microRNA 203 targets the BMP antagonist BMPER and is activated by endogenous PITX2. So what is so important about BMPER? Well, BMPER is expressed in dental epithelium during tooth development, showcasing the tissue-specific activity it possesses. Another important microRNA is microRNA 200C 141. If this is deleted, our mouse Jerry will show enamel defects. But why? Because a downregulation of E-cadherin, which helps with cell adhesion, and amelogenin, which helps with enamel formation, occurs. After a bunch of experiments and analysis, what did they find? They saw that microRNA 200C represses noggin in dental epithelial-like cells. They also saw that noggin was present in greater levels in the labial CL, seen in green, than in the control. Also, inactivation of Dicer1 can lead to defects that deal with microRNA control of epithelial stem cell proliferation and differentiation. What else did they see? Well, they saw many types of microRNAs that were expressed differently from the labial CL and the ameloblast regions. By comparing control and a PITX2 Dicer1 knockout variations of the mice, it showed that there are 20 microRNA and mRNA interactions that were vital to dental epithelium. Particularly, 3 of these upregulated genes coded for the BMP signaling antagonist. They were noggin, BMPER, and HTRA1. When noggin is looked at carefully, it was seen that an overexpression of this caused the inhibition of ameloblast differentiation. Hence, in normal mice, low levels of noggin would be seen. Now, what did we see with microRNA 203? This microRNA can inhibit BMPER, where a decrease in BMPER levels is correlated with overexpression of microRNA 203 in LS8 cells, or the ameloblast-derived cell line. The expression of microRNA 203 can also cause the loss of luciferase activity, though if the microRNA seed sequence is mutated, there is no loss of luciferase activity. To further investigate if microRNA 200C actually regulates BMP signaling, a BMP reporter plasmid that had 2 BMP responsive elements was used in conjunction with a microRNA 200C and were co-transfected into LS8 cells. By doing this, it proved that BMP signaling indeed played a role in dental epithelial differentiation. The transcription factor PITX2 is highly expressed in incisor cervical loops, or CLs, but are only expressed in low levels in ameloblast. MicroRNA 200C expressions starts in labial CL and is expressed a lot in preameloblast. This suggests that microRNA 200C may cause the differentiation of ECAD expression, cell adhesion, and ameloblast differentiation. The green here is near the labial cervical loop, showcasing high expression of PITX2. The blue serves as a contrast and to visualize the nuclei. Finally, it was seen that in microRNA 200C 141 knockout mice had defects not only in their enamel but also in the cell adhesions they were a part of. In these knockouts, there was a defect in enamel mineralization as well as an eruption defect in the third molar. The incisors also grew smaller than usual, the dentin layer was thinner, and the surrounding bone was not that strong. This suggests that a defect in stem cell differentiation may have to do with abnormal enamel formation.